If you'd like to follow along with this PowerPoint presentation, you'll find it in the Blackboard Classroom in the Week 2 folder for the Hoxie Gorge Lab. Hey there, Bio 110 students, this is Dr. D. I want to take a little time to talk about some of the things that we didn't get a chance to talk about as to what we were doing out there in the field and why we were doing it. So we took a trip over to Hoxie Gorge, which is a nature preserve, and it is owned by SUNY Cortland. So if you get a chance to go out there and take a nice hike, I would highly recommend that, and I'm sure you'll enjoy it. So before we get started with that, let's talk about what we're going to do next week in class. So you've got your first essay, and you were sent out an email, and you're going to find the instructions to this essay on the Blackboard Classroom. There are 2 articles for you to read to get prepared for it. It's going to be an in-class essay. You're not allowed to use your notes or your articles, so you definitely need to be prepared. You're going to get 15 minutes to write the essay in the beginning of class. And then we're going to do Lab 3, which is cell size and diffusion. So read that ahead of time so that you know what you're going to be doing in class as well. And then the following week, you have your first quiz, which is a 20-question quiz, and it's going to cover the first 3 labs that we've done. And then we're going to do the Nature of Science Part 2, and you have a homework due on page 24 of the lab manual, so make sure you go ahead and do that. Let's head back now to Marathon, New York, to the Hoxie Gorge Preserve together, and remember the plant that we study. So goldenrod is a very common plant indigenous to our area, which blooms in late summer. By the time we got to these plants, they were well established and quite tall. This plant is a very important source of nectar for bees and wasps, especially since it keeps its flowers well into the fall. On the trail that we walked, we noticed tall trees to our left and an open meadow to our right, which was filled with goldenrod. Our field site was marked with pink marking tape, and our transect was marked every 5 meters with the same tape. We chose 6 groups to count goldenrod galls every 5 meters. There are 2 types of galls that we identified, the ball gall and the rosette gall. Now, ball galls are produced by the plant in response to the growing larva of a goldenrod gall fly, which is growing in the stem of the plant, and this irritates the plant so that it produces more tissue around that site. The rosette galls are produced in response to the goldenrod gall midge larva that is growing in the upper part of the stem of the plant, and this produces a flower-like structure. So if you remember, we were standing on a road that was a transition point between forest behind us and meadow in front of us. And this area is known as an edge, and edges have different organisms and habitats associated with them than the interior of a forest or a meadow. Humans can have a detrimental effect on the health and diversity of the ecosystems by introducing edges artificially and breaking up forests into small sections. So we were charged with doing an observational study of an area of the meadow where goldenrod was present and plentiful, and we wanted to see if there was a difference in the number of galls present near the edge versus at the interior of the meadow. So we chose 6 groups to count 6 areas along a certain distance from the edge using a quadrant, and the distances counted were 5, 10, 15, 20, 25, and 30 meters from the edge. And some of the observations that we made were that not all goldenrods have galls. So we asked the question, is the number of goldenrod galls different in different areas of the field? So we know that there are different organisms at the edge than there are at the interior of the forest or the meadow. So we came up with a hypothesis. Goldenrod gall presence is affected by distance from the edge of the habitat. And then you were asked to come up with a prediction and write that prediction on page 8. And so if you haven't already done so, go ahead and write that prediction down. So I'm going to do a quick review of the design of the study that we did. Keep in mind that we are not influencing any outcome by introducing a variable. So we do not want to call this an experiment. We're simply observing and counting the number of galls present at different points from the edge to the interior. And we have only 1 group of subjects and no control group. So we wouldn't want to refer to this study as a controlled study or experiment of any kind either. The materials that we used in our study were a tape measure that measured out our transect. Pink marker tape was used to mark distance from 0 meters to 30 in increments of 5 meters. We also used a quadrat, which was a prescribed size so that we could maintain the same size with each trial that we did for counting. And the quadrat was supposed to be placed down on the ground, and then when counting was done, we flip it over so that we use an adjoining section and we have no overlap with the plants that are being counted. The picture to the left is showing a grid of an area of field where you could do random sampling by just choosing certain sections of the grid and using your quadrat to count those, or you can sample along a transect, which is what we did. So our transect was divided up into 6 distances of 5 meters. So we had 6 groups, and each group went out and sampled at the various distances: 5, 10, 15, 20, 25, and 30. So we had a sample size of 6. And the quadrat was laid down in 6 different adjoining points. So we had 6 trials at each distance. And then those 6 trials were averaged together, and that gave us the total number of galls at each distance. So we had a sample size of 6 with 6 numbers of total galls. The last thing that I want to go over with you is how we analyze our data. In science, it's not good enough to just say that there's a difference in the number of galls present at each distance, and therefore I believe that the edge has an effect on the number of galls present. Now, we need a way to determine how big of a difference there is and whether or not the difference is big enough to say that some force is driving that difference. That is when we use statistics. We will use a test called the chi-square test, which is a fancy equation that takes into account how big your sample size is, and it compares the observed or counted total number of galls for each sample and compares that to what you would expect to find if any differences found were caused by random chance. Once we plug in our observed values for total number of galls and the expected values into the equation, we get something called the chi-squared value. This value is a number that tells us how big the difference is among our samples, and the higher the number, the greater the difference is. I have uploaded the class data and the chi-square values for each section to Blackboard, so make sure you record those in your manual. You can think of statistical testing as a way to place our results along a probability spectrum, with no difference being to the far left of the spectrum and a large difference is to the right of the spectrum. Our results will fall somewhere within this spectrum. The chi-square test puts a number to your results. That number gives you the probability that your results are due to random chance only. The lower the probability, the less likely random chance is the cause. You can see in the chi-square table the probability spectrum that I've highlighted here in yellow. To the left on the spectrum is a higher probability that any difference that you observed would be due to random chance. And as we go to the right of the spectrum, the probability values get smaller, and there's less likely of a chance that chance was involved, and some other force must have caused the differences observed. The gold standard in biology for an acceptable probability value is p is less than or equal to 0.05, which you can see encircled in blue. If we use this p-value, we can look up the chi-square critical value that we can use to gauge whether or not our results are different enough to be considered significant. Our sample size was 6, so what we do is subtract 1 from 6 to give us the degrees of freedom, which you can see by the green arrow is 5. And then we look up the critical value at 5 degrees of freedom, and we see that it is 11.070. If our chi-square value is below this critical value, then we can say that any differences observed in our study in the number of galls counted are due probably to random chance. And if our number is 11.070 or higher, then we can say that there is a significant difference in the number of galls at varying distances, and that some other force probably has caused this difference. We can't say exactly what that force is at this point. We can only say that there is a significant difference. It is not that important for you to know how or why we use 5 as our degrees of freedom. So don't worry yourself about understanding that. But it is helpful to understand why our gold standard is p of less than or equal to 0.05. So we can use a simple probability model using a coin toss to explain that. So I'm going to do a demonstration in class in front of you guys, and I'm going to show you that I'm going to flip a coin a number of times, and that each time I flip the coin I have a 50-50 chance or a 0.5 chance to get either heads or tails. So I'm going to flip the coin once and show you that I ended up getting heads, and that is a 0.5 chance of getting heads. Then I'm going to flip it again and show you that I got heads again. Now that is a 0.5 times 0.5 probability of happening, or a 0.25. Then I flip it a third time, and if I get heads again, that probability goes down to 0.125, that can happen 3 times in a row. If I flip it a fourth time and I get heads again, that probability goes all the way down to 0.0625. So you see the probability is getting lower and lower that I'm going to be able to do this. If I'm able to flip it 5 times in a row, probability goes all the way down to 0.03125. And you might say, hey, are you sure that there's not something fishy going on here? Do you have like a coin that has 2 heads to it, possibly? So this is where our level of comfort begins to shift, where we say, that seems like 5 heads in a row. I don't think random chance would allow 5 heads in a row. Something else must be going on here. Maybe Dr. D has a 2-headed coin. So the gold standard for biology, this p-value of 0.05, really is just based on a feeling. It's that level where we begin to feel uncomfortable that random chance is causing the changes that we see or the differences that we see. And this is where we start to believe or think that something else is going on. Something is possibly having an effect. A force is having an effect, causing the results that we see. The last thing that we want to do with our data is graph it. This will help us to see any trends that are occurring. The chi-square test can tell us if the difference we see in the number of galls at each distance is caused by chance or by some other effect. However, it cannot tell us what that effect is. Graphing the data can help us see if there is a linear trend and in what direction that trend is. You could have a positive trend, you could have a negative trend, or you could have no trend at all. For example, a positive linear trend shows us that the further out we are from the edge, the higher the number of galls we find. This finding could support a hypothesis that the edge does have an effect on the number of galls, and it's possible that that effect is a limiting effect, and the effect becomes less the further out we get from the edge. So you can see how adding a graph to your data analysis can give you a more complete picture of the data and help you to understand the findings from the chi-square test and determine whether or not you have trends leading to the possibility that we do have an edge effect on the number of galls. I'm going to go ahead and plot some data and finish up this video. You'll see me add the expected values that I used for the chi-square test just to see how the observed values are distributed around them. My data does not show a trend, and my chi-squared value was below the critical value, so I will have to reject my hypothesis that the edge has an effect on the number of galls. Take care, and I'll see you in class this week.